Very good evening, one and all. I am Arpita from the IJCP group, the moderator for today's session. The topic that we are going to cover today in our webinar is CRISPR and Hematology, gene editing for hemoglobinopathies. And to address this topic, we have with us none other than a very known hematologist with us, Dr. Prantar Chakraborty sir. It's an honor for me to welcome sir again to our platform, and though he doesn't need any introduction, but still to start with the session, sir is ex-professor and head Department of Hematology at NRS Medical College, Kolkata. He is a consultant hematologist at Zoho Corporation, Tenkasi. So with this, a very brief introduction of yours, sir, I would now request you to please start with your presentation. Over to you, sir.
Yeah, thank you. And when clinicians discuss gene editing, you'll understand that it has come into clinical practice. So today I'll be basically discussing something about gene editing and also talking about the CRISPR-Cas9 revolution. So does anyone know who these two ladies are? If you know it, you can type it in the chat box. She is from France and she is from the US, right? So it's Emmanuel Charpentier and Jennifer A. Doudna. And why I am showing you their pictures is, yeah, they obviously look beautiful, but they received the Nobel Prize in chemistry in the COVID year in 2020 for their work on CRISPR, right? So it's not very long that they have been working, and by Nobel Prize standards, they are quite young, right? So this technology has been such that the pioneers in the field have received the Nobel Prize within 15 years of starting their work, which is quite rare. So today's journey will be about what is gene editing, a bit about what is the difference between, gene editing, gene augmentation, gene correction, and then what are the different ways of editing the genes. What is this CRISPR-Cas9 technology which led to the Nobel Prize? Why hemoglobinopathies are a good target? And how is CRISPR technology curing sickle cell disease as well as thalassemia? What are the limitations? And what is the future? So let me start by telling you about gene editing. Now, gene editing means that there is an error. You are either correcting it or just removing it so that something else works. So sometimes we talk of gene therapy where we actually introduce an entire new gene and replace it. And so even in stem cell transplant, we do the same thing, right? So we ablate the defective genes and give new healthy genes so that they work. Gene editing is a bit different. Here, what we use is we use a pair of scissors and then we insert something there for correction. So there are two types of gene editing: RNA manipulation strategies and DNA manipulation strategies. For RNA, we use ASOs, which can be again different types, single, multiple, combination. You can use small nuclear RNAs or microinhibitory RNAs. And for genomic manipulation strategies, where we predominantly work on the DNA, we can use nucleases. Now, what are nucleases? These are enzymes which can cut the DNA. So the first thing is, for editing, you need to cut the error, right? And then you can put in something which will correct it. So nucleases, first generation were zinc finger nucleases and talons. Talons is still used, but what we have been overwhelmed is by CRISPR Cas9 and Cas12a. There are other nucleases also. So the first learning today is that Cas9 is also a nuclease, right? But it acts in a different manner than what we were using earlier, like the zinc finger nucleases and talents. There are certain nucleus free strategies which I'll not discuss today, but they are using base editing, primer editing, and primer nucleotide editing, so that even without cutting, you can change a particular nucleotide. So it is more specific, and there are a lot of experiments. So when I talk of CRISPR Cas9, I actually am talking about double strand breaks. Whenever you read literature, and there have been thousands of publications now, I think about 1 million publications on CRISPR Cas9, mainly by our bench scientists. So, as I told you, in the first generation, we used zinc finger nucleases. Then we were using something called talents, which would identify a tail, which is an associated ligand, and then cut it there. And now what we are using is, we are using something called Cas9, which uses PAM, right? Now you might ask me, now what is this PAM, right? Well, it is a protospace motif, right? So originally, we found that bacteria use this CRISPR Cas9. Why do they use it? Suppose there's a bacteria here, right? And then a virus comes in, like a bacteriophage. So when it enters the cell, Cas1 and Cas2, they break part of it, and they already have something. Mind you, bacteria also have adaptive immunity. So this CRISPR Cas9 is an innate part of their adaptive immunity. It's not that only we have it. So there are spaces and there are prototypes which are called differently. So like this, right? So this Cas1 and 2 can identify between viral DNA and host DNA, and then can integrate it, which then is identified by a something called guiding RNA. Now we are designing single guiding RNA. I'll again discuss it a bit. And then Cas9 is guided by this, which goes and cuts it. So, is cutting enough? No. If it only cuts and there is no repair, then obviously the technology is of no use. So, after a DSB, what we do is called DSR. And there can be two types of repairing, right? One is called non homologous conjugation, right? And the other is homologous divalent repair or di-strand repair. Now, I'll again discuss this, but remember, if you have to make it very simple, that CRISPR-Cas uses a nuclease technology, and how does it know where to cut, which is the post office, right? So that the letter is delivered at the correct address. This is spam, right? And it goes and cuts it, and then there are ends lying like this, 3 prime, 5 prime. There's a gap here. And then what we do is, using different technologies, we repair the gap. Now, the bacteria use it to integrate the viral DNA, so that there is an attack the second time, they immediately attack it. But what we are trying to do is, we are trying to introduce certain genes here which can ameliorate the defect, or knock out some gene, or put in a new gene so that it can act. So, suppose I take the example of beta thalassemia, right? So there is an aberrantly spliced mRNA, and you can use, as I told you, non-nucleus methods also, like ASOs, which can correct it, and beta globin gene production happens. So to make it very simple, this is how, say, we are breaking the road. The road is meant for my vehicle to go. So, there will be transcription and then translation. So what we are doing is, we are first breaking it through Cas9. right? And then either non homologous repair or homologous repair, you put in a patchwork. And then you expect that the road will be smooth and normal proteins will be produced. So, first thing is, what is the full form of CRISPR? Clustered, regularly interspaced, short palindromic repeats. Now, each of it is an English word. Palindromic, why? Because if you read it from 3' end or from the 5' end, it's the same. Why repeats? Because the same thing is repeated, not only in bacteria, but also in all mammalian genes. And they are regularly interspaced. So, as I told you, there will be something like this, a dotted line. You have the code, you have the space. You have the code, you have the space. And you have complementary strands here. So, when you use Cas9, the full form of which is CRISPR associated protein 9, it acts as a Caesar and it identifies the exact place where it will cut. And how does it identify that? I told you by the PAM motif, right? Now, I cannot be continuing this discussion without telling you some of the history. So, as I told you, it's not a very old technology. In the first decade of this century, it was named as CRISPR, and then the role of PAM was understood. We understood the mechanisms of Cas9 cleavage only about 12 years back. And then Cas13a RNA editing tool was there. Clinical trials had started in 2016, and in 2020, they not only received the Nobel Prize, this same technology was used to detect SARS COVID virus. And in India, this same year, we discovered something called Feluda. So some of you might have heard about it. It was developed by IGIB in New Delhi, and then it was taken up, but then it was not used much. And parallelly, a lot of CRISPR associated nucleases were being tried to make them more specific, less off targets, and also to make it cost effective. So, multiple clinical data were released in 2022, and in December 2023, it has not even been 1 year, the first products for curing sickle cell disease and beta thalassemia were approved within 10 days by the European Medical Agency and the US FDA. So, what is this CRISPR mechanism, right? In CRISP, the target DNA sequence is identified. How do you identify that? Because you know which point you want to work on. And then you have a gRNA or a guide RNA with a complementary DNA sequence which binds to the target DNA sequence. But it might also bind to certain other things, but it will only bind when it gets PAM, right? So, PAM is a name you have to remember. In the bacteria, if there is no PAM, this Cas9 does not act. In humans also, it's the same. Now, once the target sequence is reached, it binds and cuts both strands of the DNA, right? So it is cutting like this. And then, once the target sequence is cut, there are three potential results. That depends on what you want to do. You can repair it by introducing a mutation in the DNA, which, if there is a gain of function, you can knock it out. You can engineer enzymes to make cuts on either side to remove the target DNA and then join them, right? I'll show you how that is done. And the indigenous DNA repair system, right? So if you are exposed to radiation, if you are exposed to topoisomerase toxins, when breaks occur, body itself heals through the same mechanism, and we have copied it. So we use an endogenous repair mechanism and do it. Now, Cas9 was the first protein which has been identified from Streptococcus pyogenes. It is knockout, knock-in can be done. It is widely used in genome editing, but it has more off targets than Cas9 variants. So, as I told you, we are trying to develop variants that can be DCas9, Cas9 nicase, Cas12a, Cas13. So, Cas12a, we use the same number and do not use a prefix when they are from bacteria. So, acid aminococcus species, leptotrichia species, each one of them have its own advantages and disadvantages. Like Cas12a does not use a tracker RNA, which is used by Cas9. And then you do not use HDR machinery or a PAM if you use Cas13a. So that is how we are trying to develop. And let me tell you, India already has patented more than three such enzymes. So don't think that we are not in the game. So you get the CRISPR RNA, the Cas9, it breaks it, and then non homologous junctioning or repairing. One is that you use certain enzymes which block this ends, 3' ends, and then stops at a chi site. Okay. So there are something called REC-BCD or REC-A. They break it, and then through a spring mechanism, it gets a homology and it gets joined here. Okay. And here you directly do the joining. This has less errors, right? So if you think like you will work on an experiment, you would prefer HDR than NHEJ. But another question remains, right? How will you deliver this CRISPR Cas9? So there are so many methods for that. There are physical methods, right? Like micro injection, electroporation, which we use in gene therapy also, hydrodynamic delivery using water molecules. You can still use the vectors which we use in gene therapy like lentivirus or AAV, or you can use liposomes, polyplexes, cell penetrating peptides, or even gold nanoparticles. So each has its advantages and disadvantages. Let me talk of some of them. So the commonest viral vector which has been used is adeno-associated vector. But remember, more than 90% of the Indians have already been exposed to adenoviruses. So if you use this as a vector, we might use our antibodies to destroy them. Microinjection is, as the name suggests, electroporation is high electric charge, and chemical methods like lipid nanoparticles or gold nanoparticles. Now, amongst the viral vectors, depending on their size, we also use lentivirus and retrovirus. And how that viral particle acts, just like it was acting in bacteria, it goes and transfects the cell, it is transported into the nucleus, and then the vector genome is there, and the guide RNA is there, which breaks the Cas9, and then transcriptional activity happens. Once this transcriptional activity happens, this Cas9 will go and target the place where it is meant to be. So there are advantages and disadvantages of different, what we say, vectors or cargo carriers, right? So what is the cargo they carry? They carry the Cas9 plasmids or the RNAs or different types. I'll show you. But microinjection and electroporation have been used. Remember, they can cause cell cytotoxicity. Some cells are not susceptible. Lentivirus immune response low, but it can pack in only 18 kb, right? So when we send satellites to the sky, you know, we use certain rockets depending on their cargo. Nowadays, we are using drones, right, to transport blood units, to transport medicines, blood samples. So this can carry a small, adenovirus can carry 35 kb, adeno associated can carry only 4.5 kb, right? Lipid nanoparticles, nanosomal particles, They are dependent on cell type, but it depends on how the endosomes in the body will process them. So, what is the in thing? You'll hear more of this using microfluidics, which can transfect, say, around 3 million cells in 10 seconds. But this is at the experimental stage and it costs a lot of money. Now, I was trying to tell you that what would be the cargo. It can be Cas9 or the guiding RNA encoding plasmids, which will encode it once it reaches the place. It can directly carry the mRNA and gRNA. So there are issues with mRNA stability, there is immunogenicity, and this is more expensive, right? Ribonucleic protein complex can also be used. There can be fewer off target cells, highly efficient, but it is the most expensive. So, you can call them as first generation, second generation, and third generation, but all three are being used right now. So, I hope I have been able to make you understand what is the technology. Now, how do you use the technology in biological sciences? You can use it in experimental research, like doing certain functional genomic studies, generating disease models. You can even develop in silico, in vivo chip studies. Chip is, as you know, clonal hematopoiesis, right? And then in silico, you can identify target and screen for drugs, and it takes less time. How are we using it in human therapeutics? Today, the next 8 minutes would be focused on this monogenic hereditary disease like beta thalassemia, sickle cell anemia, manipulation of virus cellular target like HIV and CAR T. I can go on for hours on CAR T, but it's essentially the same kind of thing which we are doing. Now, why are we targeting hemoglobinopathies? Because, as I told you, most of the hemoglobinopathies, including beta thalassemia and sickle cell or hemoglobin E, they are all monogenic. And we can precisely edit the gene responsible for producing the abnormal hemoglobin. Or, suppose you say, no, I'll not make it that specific, be it beta thalassemia, be it E beta thalassemia, be it sickle cell. I will only increase the hemoglobin F. You can do that also. You can switch on the production of fetal hemoglobin, which can alleviate the symptoms of disease by preventing the red blood cells from sickling and can improve the oxygen carrying capacity. So, in short, beta thalassemia, the problem is point mutation. We already are using transplants, hydroxyurea, and now I'll be talking of gene editing because there are other drugs which do not actually cure the disease. In sickle cell, again, it's a single gene disorder, right? So it becomes valine at 6 position, polymerization, reduction of nitric oxide, and multi organ failure. So CRISPR Cas9 has been the most attractive proposition now, and we are trying to do it in two ways. One is by increasing the hemoglobin F level by targeting BCL11A or the promoter regions LCR, or directly change the mutation back to the wild type. Base editing, as I told you, like primer editing and others, they are also trying to convert the hemoglobin S to hemoglobin Makassar, and this has also been approved. And there are others where prime editing can change it to hemoglobin wild type. So, as I told you, there are three steps: identify the sequence, cut the sequence, and then either join them or delete something. So these are the mechanisms we are using. And for cutting, we can use ZFN, TALEN, or Cas9, which actually is being used the most today. So suppose you have a IVS15, right, mutation, which is the commonest in India. You go like this, cut it, and then intron 2 splicing mutation, put it, or you can reproduce the HPFH mutation and the hemoglobin F will be such that they are converted into non-transfusion dependent. So non-homologous end joining or homology directed repair It depends on what you're trying to do. And another is whether you want to do it in vivo or ex vivo, right? You want to put it in the human body and get it done, or you take out the stem cells, modify them, and give them back. So, beta globin gene repair is being done, gamma globin reactivation for increasing hemoglobin F, and lipid nanoparticles can do it. So, just to review, if you are watching your WhatsApp, that you identify the defective DNA strand, you create the guide RNA, we call it sgRNA, single guide RNA, and then it finds the DNA, it cuts it, and then there can be non-homologous or homologous DNA repair. So one of the first studies which was published quite back in NEJM was targeting the BCL11A, which can lead to hemoglobin F increase. And they did it in only two patients and saw that fantastic response in terms of hemoglobin F increase and also amelioration of symptoms. So I'm just showing you, don't have to go through all this, but look at CRISPR Cas based systems going on. So many companies are there, their products have already been approved, others are also there, and clinical trials are in phase 1, 2, or 3. So, what is the cargo? It can be DNA plasmid, messenger RNA, or ribonucleoprotein. I told you this is the most sophisticated. Delivery systems I've already talked, in vivo and ex vivo. So, in vivo means you give an intravenous injection and then the gene gets corrected. Ex vivo means you take it out, grow them in cell culture, and correct them. So, if you think these are all future technologies, sorry. As I told you, FDA approved the product on 8 December 2023. EMEA approved it on 15 December 2023. And these are available. But what are the limitations? Editing efficiency. Till now, the highest efficiency we have achieved is 50%. So don't think it's all or none. I edit all. No, the design of guide RNAs is still challenging, and then protein optimization of this guide in protein, because you do not know how much of transcription and translation would occur, off target effects. This is important as a safety warning. They might even activate oncogenes, and we are doing most of the studies in silico. AI is helping us, but we still need better prediction tools. So what are the challenges today that we have to overcome? So, delivery challenge, ethical issue, whose proprietary thing is that, off targeting, and then, as I told you, can it cause cancer? And then, can we target even children? These are some of the challenges. What about financial toxicity? For us clinicians, let me tell you, I told you that they have been approved. The average cost is $2.2 million, single time treatment, which comes to 18.5 crores rupees. Do you think our patients in India can afford it? But I don't want to end on a pessimistic note. So here's the good news. BIRSA 101. It's an acronym for Biomedical Innovation for Relief from Sickle Cell Anemia through Gene Correction 101. We have already reached phase 1 of this, and AIMS is one of the sites for clinical trials. It is being done by CSIR, Integrative Genomic Institute of Biology in New Delhi. And these are the facilities which are already there in AIMS. Clinical trials will start soon, and maybe we will not charge crores of rupees. We will be doing it at a much lesser cost, much less cost, at a higher efficiency. So thank you very much. I think I'll stop sharing.
Thank you, sir. I would say it was an outstanding presentation. And sir, your explanation was truly remarkable and highly commendable, sir. The way that you have used the digital pen to explain each and everything, that was really commendable. So, moving on to the questions, shall I go ahead?
Yeah, please go ahead. Did people fall asleep?
No, sir. So we are getting questions. The
Okay.
first is, what are the key challenges in ensuring the long-term efficacy and safety of CRISPR-based therapies for hemoglobinopathies, sir?
Yeah. As I told you, the major challenge is safety and then efficacy. We have not been able to cross even 50% efficiency now. And we have to avoid off target. So, as I told you, India has patented its own nucleases based on CRISPR Cas9. We hope that we will develop more such specific, and then the safety would be assured. Now, if you talk about long term, right? So, you start producing fetal hemoglobin or you correct it. What will happen after some time? We don't know. Long term studies are going on, and whether we can again use it is $1 million question. I do not know the answer. I think the biotechnologists in our country are doing a phenomenal job. Incidentally, CRISPR Therapeutics, which sells that product, the CEO is Mr. Samarth Kulkarni, right? So all these US companies are also headed by Indians, but unfortunately, the cost is high because of the shareholders. So if we do more academic studies in India, maybe we will be able to overcome the challenges quicker and get good results.
Thank you so much, sir, for answering this query. Moving on to the next one.
Yes.
It's written with CRISPR technology showing promising result. How can we ensure that these treatments become accessible and affordable for patients in resource-limited setting, especially where hemoglobinopathies are highly prevalent?
Yeah, so that's why I showed you BIRSA 101, right? It's a project funded by Government of India, and many institutes are working together. And I am proud that my friends are doing a phenomenal job. We have to develop our own technology, and it's not about the product. Every process has to be indigenized because most of them are patented and they are quite expensive. So unless we make in India and we indigenize those technologies, every step of it, be it the guiding RNA, be it the nucleases, we will not be able to bring down the cost.
Right, sir. Right, sir. And the next question is, how does the choice of gene editing targets, example, PCL11A suppression versus direct mutation correction,
Yeah.
influence clinical outcomes?
Yeah. So let me tell you, we were gung ho on doing corrections. And then we saw that we were not achieving the kind of success we were achieving it. Moreover, if we have to make it cost effective, if we increase the fetal hemoglobin by targeting BCL11A, then it can be used in many diseases. So what happens is this is all commercial also, right? The demand increases, the number of patient increases, so then there can be certain successes. Now whether ultimately targeting BCL11A will be the king or correction of the single nucleotide will be the king, I think in another 4 to 5 years I'll be able to give you the answer.
Right, sir. There are few more questions coming in.
Mhm.
This is do you foresee CRISPR being used in conjunction with other emerging therapies such as gene silencing or pharmacological approaches to enhance the overall management of it, sir?
Yeah, so CRISPR, if it works, it does not need augmentation, right? Gene silencing can be done by that. But I do agree that in the long term, if we see that the protein translation is decreasing, but maybe the thalassemia becomes a non-transfusion dependent instead of getting cured. So chances of augmenting it, and as I told you, again doing some gene editing, they can all come. So this, why I call it a revolution is because it has given us a technology which we can play with, and that's why we can use it, and we can then use other things. Now I have not gone into those things like electroporation and all, designing the cargo. is so important, even for CAR T and even for the hemoglobinopathies. So, better design of the cargo, better delivery of the cargo, and better integration of the cargo. Ultimately, our aim is to cure the disease. But if you ask me, frankly, from a public health point of view, prevention of hemoglobinopathies is more important. It is more cost effective at this point of time.
Right, so we have two more questions.
Yes.
The next is, what protocol or guidelines should be established for long term monitoring and follow up of patients who undergo CRISPR based gene editing?
Yeah, they have already been laid down in the clinical trials. You can go to the clinical trial sites and you'll be able to get all the protocols. These are not short term follow up studies. So, clinical trial is ongoing. FDA is approved based on a registration trial information. So, they are being monitored for off targeting, for other safety issues, and also measuring antibodies against this cargo.
Right, sir. The next question is, what ethical consideration should be prioritized when implementing a CRISPR,
Yeah. So, the
especially
number.
regarding germline editing and its implication?
Yeah, so here, mind you, we are not editing the germline, right? It's a somatic cell mutation, hemoglobinopathies, and we are not trying to play with the germline at present. So you can tell me, okay, then what happens to the next generation? There are a number of ways. Thalassemics today still are marrying and they are getting children. So if a thalassemic marries a non-carrier, then the children will only be carriers. So we are not playing with the germline at present. We are trying to cure the disease in that person. I think germline mutations, as you rightly said, are very important ethical issues, but there are other ethical issues. India doesn't have a guideline today so that how this editing should be done, what kind of consent should be taken, and you are dealing with a vulnerable population. So if you look at the indications of the commercially available products, it is any person who is above 12 years of age who is fit for a stem cell transplant but does not have a donor. That is the indication at this point of time.
Right, sir. One last question. I think sir has already answered this, but still I would like to read it out. What are the
Mhm.
current limitations and challenges in translating CRISPR-based therapies, especially in cases such as sickle cell disease and beta thalassemia, from clinical
Yeah.
trials to widespread clinical practice?
Yeah, even in clinical trials, we are facing challenges, and I've showed you the challenges in terms of the cargo, in terms of the delivery, in terms of the guiding RNA, and in terms of repair, whether homology repair, double strand break repair, and then obviously you may repair, but still what protein manufacture will happen, the efficiency of that is also a question. So the last obviously is off target effects.
Right, sir. Thank you so much, sir. That was the last question for today. And before concluding with the session, I would like to thank you for sharing your invaluable insights on this CRISPR. And as we all know, this was such a critical topic, but your expertise and your detailed explanation has provided profound clarity on this complex subject. And I'm very I'm very sure that those who have attended the session has been benefited, and the ways you have presented the PPT with such like dedication, it was really commendable. So once again, thank you so much. Thank you, sir.
Thank you. Thank you. Thank you.
Thanks to all the audiences for joining us, for putting up your questions. Thank you so much, all. Thank you.